Good afternoon. Welcome to the Showing Discoveries at the Joseph Carreras Leukemia Research Institute. My name is Fumichiro Yamamoto, and I am the host for today's lecture to be presented by Dr. Biola Javierre. I will give you a brief introduction of the speaker. Dr. Biola Javierre studied biology and biochemistry at the University of Navarra and later received a PhD degree in molecular biology with a specialty in epigenetics from the Autonomous University of Madrid in 2011. Dr. Javierre performed a postdoctoral training at Peter Fraser's Chromatin Dynamics Laboratory at the Babraham Institute in Cambridge, United Kingdom from 2012 to 17. During that time, she developed the promoter capture Hi-C method to study spatial temporal chromatin architecture. Since March 2018, she has been a Raman Kahal PI at the Joseph Carreras Leukemia Research Institute. In addition, Dr. Javierre is part of the BioInfo for Women program of the Barcelona Supercomputing Center. In 2019, Biola was recognized as one of the 15 international rising talents by the UNESCO and L'Oréal Women in Science Foundation. I understand she has just published another paper in a high-impact journal, Nature Communications. Congratulations, Biola. The title of her talk today is The Genome in the Fourth-Dimensional Context, Deciphering the Role of Non-Coding Mutations in Blood Diseases. You can send me your questions through the question mark button on the right side of your screen. Thank you in advance. Well, Biola, please start when you are ready.
Okay, so thank you very much. For me, it's very special that you will be here presenting me, because you were one of the group that were at the beginning of the Joseph Carreras Leukemia Research Institute when I started here. So I want to thank you also your help for starting the group that was quite very tough at the beginning. So basically today I'm going to talk about the main lane of research that we are following at the 3D Chromatin organization group, which is mainly focused on the non-coding determinants that are associated with blood diseases. So first of all, let me introduce and also thank the great scientific team that are following me here in this amazing path, right? And also, I want to thank our collaborators and also the funding agencies. So this picture of the lab is not the last version, right? So there are other people that recently joined, but here you can have an idea and to have an idea about who they are, right? So let's start. So the non-coding genome, which compromise more than 98% of the total, so has been overlooked. However, we have to think that more than 95%, right, as you can see here, of the risk variants that are associated with genetically complex diseases, so basically such as cancer, autoimmunity, or cardiovascular diseases, map at non-coding regions and remain unexplored due to the lack of an obvious disease mechanism or biological relevance. Interestingly, it has been observed that these non-coding variants cluster, as you can see here, a DNA hypersensitivity site. That's, as all of you know, is a common feature of regulatory region. So basically, we could speculate that the potential role of these non-coding SNPs and structural variants is that the regulation of the target gene that is controlled by the target regulatory element, by the non-coding determinant. And basically, a similar scenario we can find when analyzing also acquired mutations and also epimutations. In fact, it has been proven, we and other has been proven, that enhancers, which basically is a type of regulatory element that binds transcription factor and to fine-tune transcription over large genomic distance. So this enhancer is also a frequent target of germline or somatically acquired mutation or epimutations. But it's not so simple, right? Because it has been estimated that the human genome harvests up to 1 million of these type of regulatory elements. And although enhancers are easy to identify, basically according to chromatin properties such as histone modification or accessibility of DNA, their target genes are harder to pinpoint because enhancers, as you can see here, are frequently located at great distance for the target genes, frequently over other intervening genes, or can even locate it in an intron of a non-target gene. In addition, to make it more complex, a given gene is frequently controlled for more than one enhancers, and one enhancer can control more than one gene. And also, this is cell type specific and stimulus specific. So basically, for all of these reasons, enhancers cannot simply be assigned to the nearest gene, and most of the target genes in its cell type and in its stimulus condition of each enhancer remain unknown. Due to enhancers control gene activity through physical proximity, this is an important concept, right? So we need that the enhancer and the promoter of the target gene will be nearby to execute the regulatory elements. Basically, the spatial genome organization, I mean the way that the DNA is packed within the nucleus, not only play a key role in the role of enhancer in regulating the gene expression, right? But also it can help us to understand the disease mechanism of enhancer mutation and epimutations. So basically, motivated by this gap of knowledge, right, to the lack of mechanistic understanding and functional understanding of the non-coding determinants in disease, we decided to develop a method called promoter capsaicin method that briefly, identify the genomic regions, including also enhancer, that are in physical proximity with each gene promoters. So just to give you an idea about the method, so after fixation with formaldehyde, in order to preserve the 3D structure of the chromatin within the nucleus, we digest the chromatin with a restriction enzyme, for instance, a Hindi3 enzyme, and the digested ends are filling with biotinylated nucleotide. And this is critical, as you will understand in the following steps. So then the filling ends are ligated and the chromatin is this cross-link. After sonication, the ligation junctions containing a biotin, and this is the reason why I told you that's important, so we can label basically the ligation junction, that is the informative part, basically we can select these ligation junctions and we can amplify after an adapted ligation. And this product, this amplified product, basically are hybridized with a capture system that is composed for in this case, for more than 40,000 biotinate RNAs that hybridize against both ends of the Hindi3 fragments that overlap each gene promoter. It's around, we design around against around 20,000 gene promoters, right? And basically, the ligation junctions hybridized against this capture system, these biotinylated RNAs, are selected again by a streptavidin pull down step and amplify and sequence. So basically, this approach allows us to know which genomic regions are in physical proximity with each of these 20,000 promoters, because as closer as two regions are within the nucleus, for instance, if you look here, the red, imagine that we have a gene promoter, and the blue, we have an enhancer. These are very distant into the genome, but because these are close by, because of the DNA folder, as closer both regions are within the nucleus, as higher number of ligation junction containing both sequence will be generated and sequenced. And at this point, I would like to highlight that the capture system can be customized, right? To interrogate any collection of regions of interest according to the biological question. For instance, if you are interested in enhancers, you can design a capture system against enhancer, or if you are interested in some specific SNPs, non-coding SNPs, you can also design a customized capture system to interrogate the interactome of these specific regions. So basically, as a proof of concept, we analyzed 17 human primary blood cell types with our new method, as you can see here listed in this table, and defining more than 160,000 significant promoter interactions per cell types, which are characterized by a median distance between interacting regions of around 330 kbs, and we observe also a median of four interactions per capture promoter and cell types. And here we have an example of the data that we obtained with this method. So this plot here represents the interaction landscape of EMPP4B promoter, here represented by the dot vertical lane, along the 2.5 and 2. Upstream and downstream region in naive CD4 lymphocyte. And basically, it's dots that are also represented by red arcs, represent significant interactions after Chicago peak calling of the promoter capsid data. So basically, you can see here that in naive CD4 lymphocytes, this promoter is strongly interacting with these three upstream regions that are in, one of these is around 2 megabases away, right? But this interaction landscape is very different across cell types, as you can see here in monocyte or in erythroblasts. In fact, the spatial promoter-centric genome architecture is not only cell type specific, but is also lineage specific, as you can see here in this hierarchical clustering of the promoter capsid data that displays two main clusters containing the myeloid and the lymphoid cells, but also smaller clusters with closely related cell types. For instance, 3 types of macrophages or naive and total B cells. So basically, these results suggest that the chromatin undergoes remodeling during cell differentiation, and it can help to reshape the transcriptional decisions, right? So in addition, as we expected, promoter interacting regions are enriched for enhancer, as you can see here in this heat map displayed in the enrichment of enhancer-related histone modification. Especially, we can see a clear enrichment of histone marks associated with active enhancers, such as A3K4 monomethyl and A3K27 acetylation, right? And what is more important, we observe that enhancer and promoter pairs display an activity-dependent connectivity, as you can see here along the diagonal of this heat map. So basically, that means that if we define active enhancer using ChIP-seq data, for instance, in erythroblast, these active enhancers are mainly in physical proximity with promoters in the same cell type, but not in the other. And this is the same for all the cell types, as you can see here along the diagonal. And here we can see an example of this activity-dependent connectivity that in this case is focused on the HBV locus, which is only that contain the monoglobulin genes, part of this, and basically is only expressed in erythroblast, but not, for instance, in monocyte and naive CD8. So basically in this locus, there is one region that's called locus control region that basically is a super enhancer, right? That before being a super cool term, the super enhancer, it was described many years ago. But in any case, so this super enhancer, as you can see here, according to histone modification, is active in erythroblast, but not in other cell types where the HBB, the immunoglobulin genes are not expressed. And this super enhancer only in erythroblast is in physical proximity with the gene promoters, but it's not happening in the other cell type. So basically, with this analysis, we can conclude that enhancer promoter interactions are preferentially linked with the cell type specific activity of the regulatory element. In addition, promoter interaction with active enhancer generally has an additive effect on the cell type specific expression level. In fact, as you can see here, so basically as higher number of interaction at higher level of expression. So, and in fact, genes that are highly connected with active enhancer in a given cell type, for instance, in naive CD4 T cells, right? These are highly expressed in the same cell type, but not in the other one. And in other cases in which we cannot see a highly connection with active enhancer, the expression is quite low. So basically, the results support a direct functional role of the identified enhancer promoter interaction in the control of gene transcription. In the other hand, we focus on expression quantitative trilocytes, also called EQTLs, that basically are SNPs associated with the expression of a given gene. And the first thing that we observe is that promoter interacting regions, as you can see here, are selectively enriched for EQTL SNPs, regulating the same gene that the promoter interacting region is connected to. And this is happening for, and this is happening for a broad range of linear distance from the target promoter when comparing with randomized control, displaying here in this case in purple. And here we can see an example of this data. So here we have an SNP that is controlling in an opposite manner the expression of these two genes, as you can see here. And basically the promoter capture high-C data connect this enhancer with the promoter of both target genes. And in addition, as we expect, the promoter interacting regions are also enriched for GWAS variants in relevant disease or trait cells. So for instance, if we focus our attention in variants associated with autoimmune diseases in both plots represented by red color, for instance, celiac disease, rheumatoid arthritis, among others, these SNPs associated with autoimmunity are enriched at promoter interacting regions in lymphoid cells compared to myeloid. More specifically, we can see a clear enrichment in activated total CD4 cells. But if we focus our attention in the SNPs that are associated with traits that has nothing to do with these cell types, that in this case are represented by blue, and we have some examples such as bone density or blood pressure, we cannot see any kind of enrichment, as you can see here, represented by green color. So basically, these results not only demonstrate the power of promoter-centered special chromatin organization in improve our understanding of the mechanistic role underlying non-coding the mutation, right? But also highlight the needs of studying the 3D genome architecture of the relevant cell type for a given disease. So basically, if you want to interrogate some non-coding disease, non-coding SNPs associated with a disease, you need to interrogate the relevant cell type for the disease. So what is more important, so applying a gene prioritization algorithm that we developed, is possible to use promoter capsid high-C data to connect the non-coding variants with putative disease-associated genes and gene pathways. Some of the prioritized genes has been previously associated, right, with the disease of the trait, but many are new ones. And in fact, to give you an idea about the power of this methodology to understand, to discover potential disease-associated genes, if we focus our attention only in 8 autoimmune disease GWAS dataset, And we analyzed the prioritized genes after integration of promoter capsid high C data and all these 17 blood cell types. 76%, so this is a high number, 76% of the prioritized gene has not been previously implicated as a causal candidate gene for autoimmune diseases, and even 65% fall outside of known disease susceptibility regions. So basically, these findings demonstrate the power of high resolution 3D promoter interactomes to provide new insights about the genetic determinant indices. So just to summarize in this first work, basically I have combined high C with capture high C, basically to interrogate the promoter interactome of more than 20,000 promoters, right? And basically, we have observed that long-range promoter interactions preferentially link promoter and enhancers, and are very cell type specific and lineage specific. And basically, we have observed that there is a strong and cell type specific enrichment of AQTLs and GWAS SNPs at promoter interacting regions. And this confirmed the potential of promoter capsid high C data to connect non-coding regulatory variants with their putative target genes, right? Basically, with this approach, we have connected non-coding disease-associated variants to their target promoters, identifying dozens of new disease candidate genes and gene pathways. That is more important, because sometimes when we want to design a therapeutic approach, it's better to target a pathway more than a single gene. So basically, taking everything together as a take-home message, I would like to highlight that this work presents the first large-scale resource of promoter interactome for primary cells, and basically demonstrated powers to reveal insights into the genomic regulatory mechanism and gene pathways that underlie disease pathogenesis. So I'm just checking that I have enough time. So and also the most important slide of this block, right? So as you can imagine, an amazing team were involved in this project. So mainly bioinformatician and statistician. So I have no time to go one by one, but here you have all the people and the different team from the different institutions, and also the funding agency involved on this. And basically, I wanted to, so I have demonstrated that using promoter capsid high C data combined with epigenetic information for the relevant cell type, we are able to decipher the pathogenic role of non-coding mutation in disease. But what about the underlying molecular mechanism? So let me briefly explain our last work in collaboration with Soranzo and Spiakov teams, among others, that we published the last week. So basically, to answer this question, we have used natural genetic variation across humans as a high throughput perturbation experiment to understand the interplay between SNPs, transcription factors, genome regulation, chromatin structure, and genetic susceptibility to disease. In this case, to autoimmunity and inflammatory disorders. So we have been focused on PO1 pioneer transcription factor, which control myeloid cells. So PO1 binds at enhancer elements, which can be characterized by repressive or active chromatin. It doesn't matter in this case because it's a pioneer factor, and activates distant genes through the recruitment of chromatin activators and other transcription factors such as CBP beta, right? So to do so, from the Blueprint project cohort that's composed of more than 100 healthy donors that are fully genotyped, we profile in neutrophil and monocyte of multiple individuals, as you can see here in this table, by CHIPSI, basically the genomic distribution of PO1 and CBP beta transcription factor, also CTCF structural protein, which is involved in DNA looping, and A3K4-3-methyl and A3K27-3-methyl histone modification that are associated with active and repressive chromatin activities respectively. And in addition, we have also obtained high-resolution chromosomal instruction profile of gene promoters by promoter Kappa-IC, and we have also used publicly available RNA-seq and A3K4 monomethyl and A3K27 acetylation histone marks. And the first thing that we did with such a huge omic repertoire, we call PU1 transcription factor quantitative trilosyl, also called PU1 TFQTL, which basically are SNPs that significantly correlate with the differential PU1 binding, as you can see here in this example, where, for instance, the presence of a guanine in red correlate with a high peak of PU1, but not in the case of the presence of a thymidine, right? In which there is that correlates with no or low PU1 binding. And the first thing that we have observed is that PU1 QTLs in neutrophil display a bimodal distribution, right? Of the distance from the respective differential binding peaks. And in fact, 55% of these map proximal to the PU1 peak edge, and the remaining 45% of SNPs localize more distantly. In this case, over 2.5 kbs in this case. So basically that means that we have the SNP and the binding peak is over 2.5 kbs of distance. Interestingly, a significant proportion of these PU1 QTL SNPs are also expression QTL SNPs. I mean, a given SNP is able to control the binding of PU1, but also the expression of a given gene. And this basically reinforces the role of PU1 in gene expression control. So basically, to study the interplay between PU1 and other potentially related DNA binding proteins, we also perform QTL analysis for CBP beta, which is involved in neutrophil differentiation, these transcription factors, and also CTCF, which mediate long-range chromatin interaction. And in this case, first, focus on proximal PU1 QTL. As you can see here, we observe a decreasing effect of these SNPs on CBP beta binding with increasing distance, in this case represented by blue, but we did not observe this distant dependence on the CTC binding, in this case displayed by red. So basically, this result confirmed that PU1 and CBP beta bind in collaboration, right? And suggest the long-range genetic effect on CTCF binding. And here, for instance, we have just one example of one SNP here that is a PU1 and CBP beta QTL. And in addition, if we, instead of being focused on the proximal PU1 QTL, we focus on the distal PU1 QTLs, these distant ones are mainly cell type specific, as is demonstrated by the non-shared genetic effect of these SNPs between both cell types, the neutrophil and the monocyte. Basically, here displayed by the different slope observed in this plot. So, on the other hand, given that PU1 is a pioneer factor, we expect that the allelic effects of its binding might influence the local chromatin state. So to verify this hypothesis, we also call histone modification associated QTLs for the active A3K4-3-methyl, A3K27 acetylation, and A3K4 monomethyl histone marks, but also for the repressive A3K4-3-methyl mark in neutrophils, and we overlap this data with PU1 QTLs. So the first thing that we observed was that variance effects on PU1 binding and A3K4-3-methyl tend to be in the same direction, right? But it was in the opposite one for the A3K27 method. And interestingly, there was a pronounced bimodal distribution of distance between PO1 binding peaks and the location of the A3K4 methyl here, as you can see here, displayed by the purple color. So, given that A3K4 methyl is marked, it's associated with active promoters, this observation highlights the potential long-range regulatory effects of PO1 binding to distal DNA elements on promoter activity. So, to confirm this observation, we use our promoter capsid data to identify promoter interacting regions containing SNPs in heterozygosity that exhibit allelic imbalancing interactions. And as you can see here, this is an example. The presence, for instance, of an adenine, but not in the case of the thymidine, is associated with the DNA looping. So basically, we observed an enrichment of PO1 and CTCF-QTL within these heterozygous SNPs involved in the imbalance interaction in neutrophil and monocytes, as you can see here. And the same is true for the histone QTLs, although these are not displayed in this slide, right? So promoter interacting regions are enriched in neutrophil-specific PO1 binding and enhancer-associated A3K4 monomethyl and A3K27 acetylation mark, as you can see here. So basically, we observe these three QTL, these two histone QTLs and the PO1 QTL, were positively correlated with the level expression of the gene that they contact in neutrophil, as you can see here. But in contrast, CTCF binding at these promoter interacting regions does not correlate with target gene expression. And this is expected given the constitutive nature of many CTCF-mediated chromatin interactions. And finally, we compare the effect of SNPs that are distal, share PO1, QTL, and also expression QTL mapping within and outside of promoter interacting regions. So PO1 QTLs mapping to promoter interacting regions, here represented by dark blue, showed significant larger effects on the expression of genes that they control, compared with distant match SNP, in this case in gray, that did not map to a promoter interacting region. And this demonstrates that the physical enhancer promoter contacts mediate the transcriptional effect of PO1 binding. And here, surrounded by the rectangle, you can see an example of a SNP that significantly controlled differential chromatin interactions with the LRRCHC promoter and the region containing the SNPs, so this one, but also controls H3K4 methyl and H3K27 methyl deposition at the LRRCHC promoter and PO1, CBP beta, and CTCF binding. And also, this SNP control LRRCHC gene expression. So these and many other examples demonstrate coordinated genetic influences on PO1 binding, chromatin activity, and the formation of promoter interactions in the regulation of neutrophil gene expression. And finally, we look for evidence of a functional interplay between our identified transcription factor binding and the shaping of myeloid traits. And we observed that enrichment of SNPs associated with full blood count within PO1, CBP beta, and CTCF bound regions in neutrophil, grouped by these reds, by a red circle. So basically, we expanded this observed enrichment of SNP, and we further investigate whether our transcription factor QTLs potentially influence not only traits but also human health. For this, we were focused on 24 G1 associated studies for autoimmune disorders, allergy, and infection for the UK Biobank. And we observed that 26 proximal and 53 distal transcription factor QTLs share a genetic signal with at least one GWAS locus. And basically, using this approach, we were able to identify 27 high confidence target genes that could be potentially implicated with autoimmune disorders, allergy, and infections. And just to finish here, I want to show you an example of a P1 QTL SNP here that is associated with neutrophil and monocyte cell counts, which this SNP is connected by the 3D genome organization with CPEB4 gene promoter, and this gene basically encodes for a cytoplasmatic polyadenylation protein that basically binds poly-A tail of the poly-A tail of the RNAs to regulate the translation, right? So interestingly, this SNP also controls PO1 binding and CBP beta binding, and also the deposition of trimethyl K4 histone 3 and acetyl K27 histone 3 histone marks. So just summarizing, I would like to highlight this take-home message. So here we have a genome-wide profile on primary monocyte and neutrophil of a cohort of more than 100 healthy donor, fully genotyped. Basically, the binding of PO1, CBP beta, and CTCF, and also the genetic distribution of activating and repressing histone marks, also the gene expression and the promoter interaction. And using QTL analysis, we have shown that variants associated with differential PO1 binding underlie genetically driven differences, not only in cell counts, but also in the susceptibility to autoimmune and inflammatory diseases. And basically, this work highlights a coordinated effect of PO1 binding variants, not only on the local chromatin states, but also on the enhancer promoter contacts, and also downstream gene expression, and allows the interpretation of 27 genes that could be related with immune traits and immune diseases. So basically, all these results demonstrate that the function of PO1, the role basically of PO1 and its target enhancers in neutrophil transcriptional control and immune disease susceptibility. So yes, I just want to finish. I don't have time to go one by one to all the people involved in on this work. I wanted to highlight the huge effort from of Stephen Watts, right? Who was the first author of this paper, who made an enormous work. It has been a tough paper. It took almost 2 years during referring processes, but we are very happy that at the end the paper is out, and of course also the great help of Spivakov and Soranzo group from the Imperial College and the Sanger Institute, and among many other people, including Mattia Frontini team, also Peter Fraser, and many others. So basically, I just wanted to tell another thing that I almost forgot. So we are recruiting. We have open position for a PhD and a postdoc with computational background, but we will open also new position for PhD and postdoc position for also wet and dry researchers. So if you know that somebody that could be potentially interested, please let them know to contact with me. And that's it. I just want to thank you for your attention. I know that this is a tough seminar full of QTL analysis, but I hope that you enjoy a little bit, and I will be very happy to answer your questions.
Thank you, Biola. Very exciting development. Congratulations. Let me start with my own question. So you have the physical interactions, and then that interaction was found differentially in different types of cells. But to me, I am not sure, which is a cause and which is a result. To me, gene expression is very important because that characterizes the cell types. And then if you analyze different cell types and then chromatin structures, bioinformatics data analysis, I can understand Jesse's association. But in the experimental system, can you, propose with the result you obtained to make some specific, animal model system or?
So basically, I think we have to highlight one thing. We are working with population cells, right? So if we see that there is a significant interaction on a given cell type, but not in the other one, it doesn't mean that in all the cells from a given cell type, the interaction is occurring, right? So at the end, of course, the different cell types have different transcriptional profiles. And basically the chromatin properties and also the structure reflect this fact, right? And of course, if we have the non-coding alteration at this can affect the architecture and also the regulation of this.
One question from the audience: what is Chicago peak calling?
So basically, Chicago is a pipeline that was developed by Spivakov group, that basically allow us to call what is a significant interaction out of other interactions that are not significant, right? So at the end, we have to we after sequencing the library, we have many DIE tags that link to different positions within the genome, and basically we need to know which reflect a real interaction or which no, because basically we have to face one limitation, that is the interaction profile along distance. So of course, two regions that are close by within the linear genome, we're going to obtain DIE tags that connect both, right? So, but it doesn't mean that there is an interaction frequency. We need to see that there's more that we should be able to detect more DIE tags than expected by the distance, the genomic distance. So for instance, Chicago takes this in account, right? So basically it's a pipeline that allows us to do this calling, and it's very complex because it takes a normalized weight discovery procedure, right? That takes in account also that the full discovery rate that changes also with the distance. And if somebody wants to have more details, so the paper describing the pipeline was published like 3 years ago, something like this. And it's very, very well explained, and it's also available in GitHub.
A question for the first portion of your talk. In the hematological cell types clustering, you observe that the mono and macrophages is closer to erythroblast than granulocyte. How do you explain this, and what implications have?
So I cannot so I can speculate, but I cannot say anything. So basically, using for instance RNA-seq, it has been used to somehow reconstruct the hematopoietic tree. And this is another layer, right, to try to define a little bit more the hematopoietic tree. We are just looking for similarities. So according to chromatin architecture, it means that in this case, monocyte, for instance, is closer to macrophages. But of course, I'm not talking about that in this case, we have a lot of, so we have, of course, the monocyte give rise to the macrophages. But in other cases, maybe it's not so clear, and maybe we don't have a clear progenitor cell. We cannot say this, right? We can, at this level, we only can say that they're thinking about the chromatin organization, these are close by. And it's not only gene expression. So it's taking a lot of, it's taking just many features of the cell which reflect this. So with RNA-seq, you get some hierarchical. With histone modification, you get another one. And depending on the histone marks, for instance, you're gonna try to clusterize according to more gene expression. For example, active enhancer. With RNA-seq, you are able with this. With a structure, with chromatin structure, it's not only gene transcription. It's not only active transcription. We also have a structural interaction. We also have maybe, and it's something that we are also interested in. Is there any interaction that's repressed expression? For instance, interaction with a region that is enriched in H3K27 histone mark also reduce the expression level. So again, it's very difficult to speculate about the result that we can obtain. I just can't say, the only thing that I can say is that we need to do further validation in order to see the ontogeny, and we can only say that the chromatin architecture centering promoters is what we consider.
Okay, this is for the last question to the last slide. You have shown a SNP that controls chromatin architecture and also P1 binding and LRRC8C expression. But why is this gene relevant in neutrophils and autoimmunity?
So do you mean LRRC8C gene?
Yeah, yes.
So this gene, so it was very interesting when we find out this, because this is encoded for a volume-regulated anion channel cell unit, which is important basically to control the cell size, right? Basically by controlling the movement across the membrane of chloride ions and other organic osmolytes. What was very interesting is that this gene is strongly upregulated in terminal differentiation of neutrophil, but it was not known anything about this gene associated with this cell type. So basically, according to our results, the role basically of these SNPs, of this specific SNP in and also the role of this gene in neutrophils, we could think that maybe this gene, the LRRC8C gene, could be involved in neutrophil phase decision and function, but and also because this SNP associated with this gene is associated with autoimmunity and inflammation process, we could think that also this gene could be implicated in this pathogenic processes. But of course, we need to do further validation. This is just some observations. These are quite robust because of the statistics that underlie the analysis. But at the end, it's very important to do a functional validation, and at that point, we will understand exactly which is the role of this gene.
So you obtained many candidate genes, and then you select this one. Maybe some of them are already known to be involved in autoimmunity. And then, of course, you want to pursue the novel ones. Mm-hmm. But so just depending on the strength of the intensity or intensity of the interaction, or with additional information, you are interested in GS by GS, there is a lot of space for bias to select the ones. How can you do that?
I completely agree. So we, in both works, we have generated a list of candidate genes that many of these, so let's say that 3/4 are novel ones, but it doesn't mean that these are real ones, right? Of course, there will some ones that won't be real, and we need the validation. Of course, we can also to include other additional layers, right, to improve the analysis. For instance, we can include epigenetic data, because this analysis just perform using structural data. So maybe also the next step, also doing some epigenetic profiling or even a filtering based on epigenetic information, we can also reduce the list of candidates. But yeah, that's it. The important is that we need to know which are our limitation, and it doesn't mean that, for instance, the LRRC8 gene is key gene for autoimmunity. No, it's a potential gene to be explored in a further study, mainly for other groups that are more focused on autoimmunity or in neutrophil biology.
Next question. Is there any drivers known for chromatin looping, and also how epigenetic drugs affect these structures?
So, of course, if you change the chromatin, the chromatin properties, thinking in the epigenetic properties, it can affect the structure, right? Because so we have to think that, oh, sorry, I have a call, bad timing. So basically you have to think that, sorry, I was lost, sorry. But of course, if you change the epigenetic profile, you change activity, and you also change how is the DNA accessibility. So of course, we are starting learning about what is the mechanism that mediate DNA looping. So we know about CTCF, coenzymes, but also there are several evidences that transcription factors are important on this, right? So of course, if you change, you have an active enhancer, but you change the epigenetic profile and you move from active to inactive enhancer, maybe this transcription factor won't be able to bind. And if it's important, this transcription factor to the DNA looping, in this case, the epigenetic is gonna affect the chromatin organization. And also think on the other important thing that we have to consider that with the 3C-based methods, for instance, high-C and promoted high-C, we have to be very careful when we select the restriction enzyme to digest, right, the chromatin, because there are some of these that, for instance, are DNA methylation dependent, right? So, and in this case, it's not that the epigenetic changes genome architecture, is that the epigenetic is going to introduce an artifact in the analysis of the results.
The next question, maybe you may have already answered, but have you tried to study the genetic perturbation of P1 binding with phagocytic disorders, or alternatively with eosinophil associated disorders? So,
we didn't explore these traits. Maybe there are different reasons why. So maybe we didn't find out. So you have to think that we use publicly available GWAS datasets. So we were focused on UK Biobank, and maybe these GWAS datasets were not available, or maybe we didn't consider to include it. But I don't remember that we did it. So I think that because we considered that was not relevant, we were expecting that, you know, neutrophils more like inflammation and autoimmunity more than eosinophils and these things. But it can be done. It's just a question of getting a high quality GWAS data set, that this is not so easy, not so trivial.
Can I ask the last question?
Sure.
Apparently, your career started very well with development of this promoter capture IC technique.
Mm-hmm.
And do you have any plan to develop different types of techniques which may be very useful in understanding and promoting?
I have to highlight that I was not the only person who developed the method, so it's important also to give the credit. We were at the moment that I joined the Peter Fraser lab, there were this idea, And of course, this effort was huge, and there were many people, for instance, Mikhail Spivakov developed a pipeline, but also there were another group that also developed a pipeline. Also, Stefan Sefender was involved into the development. So first, it's important to give the credit to everyone. And of course, I love to to move forward into the methodological breakthrough. So one thing that we have already developed, it's not exactly any method, it's like the next step. So promote a capture IC or any capture IC is super cool, but of course this methodology has limitation, and for me one of the most important limitation is the cell number. So for generating a high quality promoter capture IC library, we have to start thinking about over 40 million cells. So if you want to analyze primary cell types, good luck, right? Because I think that most of these we have already analyzed. But you know, most of the primary cell types are rare cell types, so it's not possible to obtain, for instance, 40 millions of hematopoietic stem cells. So basically, this was one of the main aims on the lab. So we have developed like a low input capture IC method, and I want to say that we are super excited because we can generate with less than 500,000 cells. So we are talking about almost 1% of the initial amount. We are able to generate a highly complex libraries that, of course, we cannot detect the same number of significant interactions, but we are close by. So basically, I think that we can go down to 100,000 cells, and at that point, and already we are there. And at this point, we are enjoying analyzing really very cool cell type that gonna provide, I hope, very interesting insights.
Very, very good. Thank you so much, Biola. And also, I'd like to thank the audience.
Thank you. Bye-bye.